We are recording, taking attendance. I'm only going to mention people who are physically here. So if you're online and I say your name and I say you're not here, don't worry about it. Just go about business as usual. Ashley is not here. Barrett is here. Trahan is not here. Tickets is not here. Inca is here. Isaiah is not here. Haley is not here. Lucas is not here. Marley is not here. Noah is not here. Savannah is not here. Mia is not here. Edwards is not here. Wilkes is not here. And Trahan just came in. Just as a reminder, you only get half credit if you come in late. Of course, you can always later watch the video and send in the words and do all that to get the full credit. But yeah, gotta be here on time to get credit. Still, no one's late yet because it's not 8. So I'll start off with reminders instead of announcements. Of course, it says announcements up there. But anyway, again, you can still retake exam 1 and 2 just for the practice of it. But it won't change your grade. But you can meet me online to get all the correct answers. I can explain why they're correct, explain why your incorrect answers are incorrect. So that'll help you prepare for the final, and you'll get a 5% boost to exams 1 and 2, whichever one we meet for, or both, if we meet for both. Good morning, Noah. Always here. Again, you can still retake exam 3 until April 27th. I highly recommend it. I'm still shocked about how many people did not retake exam 2, considering how bad it was on average. So you have nothing to lose. I don't know why you wouldn't retake it. Anyway, yeah, and then after April 27th, it'll be just like exams 1 and 2, where you can still retake it for the practice, but it won't change your grade. And you can meet me online for a 5% boost and to get all the correct answers to exams. So any questions about exams 1, 2, or 3? Okay, again, the final will be in person here on May 4th from 8 to 9:50. So we're going to meet at the same time as we always do, except it's going to be extended a little bit longer. Make sure you bring a computer and a charger. Actually, probably something to write with too, and a basic calculator if you think you're going to need it. I'll provide scrap paper, but you can't use your phone as a calculator, right? Because that's how I know you're not looking something up. So as I've been saying, preaching to the choir because you're here, but some people have not been showing up for the exams. So you've got to be here. And if you know you can't make it, or even if you just don't want to make it, maybe you're not a morning person, the final's a big deal. You're like, I don't want to do that. Contact me. We'll make prior arrangements, and then you can take the exam some other time. As long as we can meet online, I can proctor your exam. And I don't mind taking it, mind you taking it another time. And I don't mind extending the time. So any questions about exams in general? Okay, sorry, I'm going to be repeating this every day until the end of the semester. So I apologize in advance. So to remind you, today's lab is 100% virtual. I will be in my office and available to help you. So even though it's 100% virtual and we're not meeting at all, I will be there if you need to meet me. So you can come to my office and meet me in person, or we can meet online, whatever you want. Let me think. It's the ecological footprint lab. So the biggest mistakes I see for this lab, just so you know, are, let me think. But yeah, think about your hypothesis questions. This is one of the few times when you can get a hypothesis question wrong. Because say, for example, and you're going to learn about it if you haven't looked at it already, and we haven't talked to you yet, but you'll learn that the average American has a larger ecological footprint than the average person on Earth. Therefore, if you say you think your footprint is the same, for example, as the average American, then when you answer the question about the world, then it must be larger than the world, right? By definition, if your footprint is the same as the average American, it must be larger than the average world citizen. Likewise, if you say your average is the same as the U.S. average, and then later start asking you some questions like, when do you think the Earth overshoot day will be? And exactly how big do you think your ecological footprint is? Make sure those questions match your answer, right? So if you think your ecological footprint is the same as the U.S. average, then all those other questions, when they're asking you for numbers and dates, it should be very close to the U.S. average, and obviously not closer to the world average. So I hope that makes sense. If not, I'm not going to spend any more time talking about it because, again, I'll be there if you need me. Oh yeah, and when you do graphs, remember the graph title always starts with figure 1, colon, and then describe the data. That's another big mistake, or common mistake. It's not a big mistake because it only costs you 1 point. So any questions about lab this week? All right, next week's lab is a free lab. Meaning, remember I told you I can't guarantee you I'll be able to grade any of your late work, grade any of your corrections, but this is an exception. So next week, choose whichever lab you want to do, redo, right? And this send me an email and say or if you redo it, you don't necessarily need to send me an email, but let's just do that anyway. Send me an email and say, okay, this is the lab that I want you to regrade. And that's it. And I will do that. It's really just that simple. So this really goes to show too, like you can either take the easy way out, like if you have a good grade or you don't care about your grade, you can do like, just redo this lab, the one that you got a perfect score on. We go, okay, so there's another perfect score. Or you could fix your grade, right? And take the time and effort to pick your worst lab, or if you missed a lab, choose it and do that. But again, just like next week, or just like this week, it's completely virtual, so we will not be meeting. But if you're doing that, if you're going to redo an old lab or do one that you missed, you're probably going to need my help. And I will be here to help you. I'll be online, I'll be in my office. And maybe you forgot to do the biological molecules lab. We can meet online. I can give you that data again, and I can explain to you what the data means so you can do that lab. So please take advantage of that. I'm trying to help your grades. Any questions about labs? Okay, and again, before we move forward, when you think about it, today's lab, in a sense, is the last lab. Because after that, we've got that free lab, and then we're done for the semester. All right. A new thing I've been saying it on, well, I haven't been saying it enough. I'm going to send out an announcement soon. That's a misprint. I will give you 20 points of extra credit if you can prove that you are registered for summer semester or the fall semester, or that you registered for graduation or applied for graduation. So just send me a screenshot that shows that you're registered or that you applied, and that's it. 20 points, not 2. What else to say about that? Yeah, make sure it's the actual screenshot that shows you're registered. So, sometimes when you go to your advisor, you'll get an advising sheet. That won't count. That just means you were advised. I want to see that you were registered, and you get 20 points of extra credit. Okay, and that's it. So then normally, or the normal stuff, I'll remind you of participation. Really quick before I do, let me write down that Edwards came in. Now we are up to date with attendance, and we're going to do things the way we normally do. I'm going to say words. You're going to write them down, email them to me when it's all over. Of course, if you're online or watching the video, you're also going to take a screenshot when I say the words. And if you're watching the video, like or comment on it and make sure you have a screenshot of that. So any questions about any of this? All right, so the first word for attendance is not a word at all. And I want you to just think about it for a little bit. And even though it's the first word for attendance, if you think about it long enough, maybe it'll be listed last on your list, and that doesn't matter. Anyway, that's a question. First word for attendance is a question. I'm going to talk about if you would like to meet sometime, we don't have much time left in the semester. There's any place you would like to meet instead of here in Hamlin at 8 in the morning. If you would like to meet somewhere else, let me know. Like, where would you like to meet? Then preferably 8, but if we wanted to, we could postpone it, like I'll meet for lunch somewhere. So let me know those. Or if you would not like to do it, or you don't care, whatever it is, answer that question. Don't care. Let's meet here, or let's not do it. I want to make sure we're meeting here, whatever the situation is. So that's your first word for attendance. Any questions? Okay, let's talk about analyzing gene pools. So again, we're talking about the evolutions of populations. This should be a little bit of a review for you. This is something you already did in that one goldfish lab. We analyzed gene pools. So let's go through this quickly. As always, I remind you, let me know if I'm going too quickly. We've got a lot of catching up to do. We don't have much time left in the semester. So imagine a wildflower population that has 2 varieties of blooms that are different colors, right? So we can see here's a population, meaning it's the same species, right? They interbreed. We have red flowers, we have white flowers. And just to remind you, not for evolution, but to remind you about the stuff you've just been tested on and you can retake the test. This is probably a situation of complete dominance as opposed to incomplete dominance or codominance, right? Because it's either red or white. Anyway, the allele for the red flowers is dominant to the allele for the white flowers in this hypothetical situation. And the genotype frequencies can be calculated from the allele frequencies if the gene pool is stable and not evolving. Now that part in the question mark is a very important part if we were really studying this at a higher level. But for a 100 level class, we're just going to assume when we do these calculations and have these conversations that the gene pool is not stable and that it is evolving. Because as far as we're concerned, just so you know, in real life, basically things are always evolving. Now it gets deeper than that. For us, again, we're going to oversimplify and we're going to assume when we talk about these things that the gene pool is always stable and not evolving. So that brings us to the Hardy-Weinberg formula. You will have to do some basic stuff with this, just like you did in the lab, but nothing too crazy. I'm going to give you some pretty crazy examples, but the stuff you're going to do is going to be pretty simple. But I'll also give you some simple examples. But this is the formula. And for those of you who have taken algebra, this formula should look familiar to you. This is a very normal formula, especially when you're working with factoring. Anyway, p squared plus 2pq plus q squared equals 1. This is used to calculate the frequencies of genotypes in a gene pool for the frequencies of alleles. And that is the official wording. This is what all the textbooks say, the frequencies, the frequencies. But in my brain, when I was learning this, it helped me to think of other words, like another way to describe it. You could also call it the ratio, right? So it's the ratio of how many homozygous dominant there are in the gene pool, how many heterozygotes there are, and how many homozygous recessive, right? It's the ratio, or you could call it the fraction or the percentage, right? All these words are technically different, but if you're just looking at the big picture, that's okay, right? We're just talking about what portion of the population is this versus this versus this. So here we go. And then there's another one. That's the more complicated one. And the other one, which we also used in the Goldfish lab, is this one. P plus Q equals 1. And again, we're talking about frequencies or fractions or ratios or percentages, however you want to look at it. Of course, the one we're using the number 1, technically it is a frequency, because what all this means is, that's the number of the frequency of dominant alleles and the frequency of recessive alleles in a gene pool. So they always have to add up to 1, right? Same here. When we're talking about genotype frequencies instead of allele frequencies, they always have to add up to 1. And some basic math you might get, right? So if I say something like, I don't know, 75%, the question would be a lot longer, but it would end with something like this. 75% of the population of the gene pool is the dominant allele. So what percent is the recessive allele, right? And you would just hope that that's just basic math. Yeah, 75 and 25, that equals 100, right? 100%. Of course, I could also already use like that, right? So that's the kind of simple questions you're going to get. 75% is this, 12% is that. And you could also get one, again, the genotype frequencies looks more complicated, but it's not that much more complicated. Again, I might say something like, I don't know, 10% are homozygous dominant, 20% are heterozygotes. What percent are homozygous recessive? I have time to wait for a volunteer to answer that, but I don't, and it's just 70%, right? 10 plus 20 equals 30%. So what plus 30 equals 100? 70. So that looks like a 76, but anyway, you're going to have those are the basic kind of questions you're going to get on the exam. So are there any questions about that? You'll also have questions that simply even easier. It'll say something like, okay, here's the formula. What does p squared represent? It's like, oh yeah, that's homozygous dominant. Or what does 2pq represent? Heterozygous. Or what is recessive homozygous recessive? Or excuse me, what does q squared represent? Homozygous recessive, right? So you need to know both of those things for the exam. Those will be 2 separate questions. There's a basic math question and also, what do these things represent? So any questions about that? Okay. Let's talk about population genetics and health science. I'm not going to ask you any questions about this. And I think this one's only from the old textbook and not in your textbook. And I'm only including this because you might be thinking, well, what does this matter? How is that even useful? The Hardy-Weinberg formula. So that's basically what this is. I'm going to give you a really complicated, relatively more complicated example, but I'm going to go through it quickly. I'm not going to ask you any questions about it. And you're not going to have any math questions that are this complicated. I just want to show you how this formula is actually used in real life. So the Hardy-Weinberg formula can be used to calculate the percentage of humans with an allele for certain inherited diseases. So we're going to use PKU as an example. Not that I'm going to ask you any questions about PKU. The PKU is a disease where you have the inability to break down a certain amino acid, phenylalanine. It's caused by a recessive allele. So here's what people figured out with health science and the Hardy-Weinberg formula. All we knew was that 1 out of 10,000 babies in the U.S. were born with this disease, right? So all that tells us is let me think about this. All that tells us if we know they have it, and again, it's a recessive allele, so that's like saying 1 out of 10,000 babies is homozygous recessive, meaning we know Q squared, right? Because P squared is homozygous dominant, 2PQ is heterozygous, and Q squared is homozygous recessive. So what we can figure out from this then, because what we really want to know when you think about it is how many heterozygotes do we have, right? Because those people, because it's a recessive disorder, they don't have it, but they're carriers. So it would be good to know, like, how many people are carrying this disease in the country. And we don't have to go test everybody. We can use good old-fashioned algebra. So here's how it works. And again, I'm going to go through this quickly. So if this doesn't make sense to you, I'm sorry. You don't need to understand this. But if you are curious about it, we can talk later after the class, and I can explain it slower. Again, we know that 1 out of 10,000 babies has this. That means we know what Q squared is. So Q squared equals 0.0001. That's just another way of writing Q squared equals 1 out of 10,000. You with me so far? 1 out of 10,000 is written as 1.0001. If you were to take 1, divide it by 10,000, that's the number you get. Now we're going to start solving for letters, right? Algebra. First thing we want to do is get rid of the square sign. So even though you don't see it on here, we square both sides. Excuse me, we take the square root of both sides. Square root of Q squared is Q. Square root of 0.0001 is 0.01, right? So now we know what Q is. Right, now here's where it gets a little bit more complicated because I kind of skipped some steps. Well, we know what Q is. Now let's figure out what P is. And we know that P plus Q equals 1. But we're trying to solve for P. So let's subtract Q from both sides, right? So P equals 1 minus Q. You guys see how I did that algebraically? You do it to one side, you have to do it to the other. There we go. Now we know P equals Q, or 1 minus Q. So now let's just plug it in. Well, what is Q? Right? So that would now be P equals 0.0, oops. Yeah, that's so sloppy. I'm sorry. 1 minus 0.01, which gives us this 0.99. So you can see I skipped some steps, but I drew them all everywhere. So now we know P, right? We know Q, we know P. We put them together. 2 times P times Q gives us 0.0198, which is about 2% of the population. So again, sorry if I went through that pretty quickly, but the point is, just by knowing how many people were born with this disease, then we can figure out how many people are carriers. The beauty of algebra. And I used to hate math when I was younger, but once I started learning it's how useful it is, I started to love it. Anyway, sorry, any questions about this before we move forward and leave Hardy-Weinberg in the past? Hey. Oh, you know what, for extra credit, you can look up, and obviously Hardy-Weinberg, these are named after people, so if you want to look it up for extra credit and tell me who these people were or how they came up with this formula, feel free. Okay, let's talk about microevolution. Once again, this is a refresher for what you learned in the Goldfish Lab. First of all, a non-evolving population is in what we call a genetic equilibrium, which is also known as a Hardy-Weinberg equilibrium. I just want you to know that this exists because, again, this is 100 level non-biology majors course. So we're really skimming the surface and we're really oversimplifying things, including this stuff. But I just do want you to know what I'm about to tell you does exist, right? So a gene pool remains constant from generation to generation when it's at genetic equilibrium. And in a sense, just so you know, this is basically like an academic hypothetical. Mostly this does not happen. And we don't have time to talk about why, and you don't need to know about why, but there you go. And when this happens, when the gene pool remains at equilibrium, another way of saying that is the frequencies of alleles and genotypes don't change, right? So that's all that's saying. When something is at Hardy-Weinberg equilibrium, we're saying that the gene pool is not changing, not for any gene, right? And the lab we did, it was only presumably one gene that controlled what color they were, right? But I'm talking about across the entire genome, they're saying nothing changes, which obviously is not true, right? At the very least, you have genetic drift, which we're about to talk about here in a second. But because generation to generation change and allele frequency is evolution on the smallest scale, this is also referred to as microevolution. And again, we've already talked about this in the lab, but just to point out again, evolution is not always big changes, right? Evolution is always not speciation where you create new species. or even when you create new phenotypes, right? Sometimes it's just a matter of, when we're talking about microevolution, what are the percentages? How are they changing? I use the same example I've used before, and I'm making these numbers up. Let's say 60% of the population, the whole entire human population has a chin dimple and 40% don't, right? And then let's say in 100 years, I don't know, 70% of the people have chin dimples and 30% don't. That's microevolution, right? We haven't created any new phenotypes. We haven't created a new species. It's just the frequencies, the ratio, the percentages that changed. So let's talk about that. These are the ways that things evolve, either whether it's microevolution or evolution. And again, these are things you've learned about, scratch the surface with the Goldfish Lab, and we're going to dig a little bit deeper now. All right. So what mechanisms can change a gene pool? Obviously, we know about natural selection. We've been talking about it the most. It's the biggest one because that's the one that leads to adaptation. It's important. But there's other ones. And again, you've tested them in the lab. We're going to talk about them here in a second. Any questions so far? All right. The next word for attendance is going to be just these initials, JSM. Today is their birthday. It would have been. For extra credit on a separate email, you can look up and explain how that person would be relevant to our course. So forget your sources, a link to your sources if you do that. But yeah, let's talk about these mechanisms of evolution. And just like in the lab, the first one we're going to talk about is genetic drift. And honestly, I was disappointed. I was just wondering, I wish I could have gotten to your brains when I was reading your hypotheses for genetic drift, because genetic drift is a change due to chance. So as the lab explained to you that when we did this simulation, there was no advantage to being white, no advantage to being orange, no advantage to being mixed. There was no mutations changing one allele from the other. There was nobody immigrating in or immigrating out. So in my mind, a good hypothesis would be there would be no change. Remember, that was an option, but very few people chose that. But anyway, let's talk about genetic drift here. Again, a change due to chance. Here's an example. This is from the old textbook. Here we are with the same wildflower population again. We've got red flowers. We've got white flowers. We've got homozygous recessive. We've got some homozygous dominant. We've got some heterozygotes. And in this population, there is no advantage to being red. There is no advantage to being white. There is no advantage to being homozygous dominant, homozygous recessive, or heterozygous, right? There's no advantages or disadvantages to this. So this is wildflower population, and by chance, these are the flowers that happen to get pollinated, right? These are the ones that reproduce by chance. So this is where we start with generation one. 70% dominant, 30% recessive. When we talk about alleles, not phenotypes. So again, those are the ones that reproduce. So then we go to the next generation, right? So those that you produce this generation, right? So again, these in the boxes created this generation, and now this generation happens to be 50-50, right? Half dominant, half recessive alleles. Again, by chance, this generation gets reproduces, and by chance, these two are the only two flowers that get pollinated. Those are two of the only ones that reproduce. So then by the third generation, the white alleles are extinct, right? And completely by chance. That's genetic drift. No advantages, no disadvantages. It's just the way it happens, right? And that is how it works in nature. Just because something changes more towards one phenotype or the other does not necessarily mean it was natural selection. Of course, that would take some studying, right? So if I just gave you this information out of context and you didn't know we were talking about genetic drift, if I just said, hey, somebody studied these wildflowers for 3 generations and these were the results, what do you think about it? Hopefully you guys would think, well, let's look into it. Like, let you know now that it's genetic drift. But again, push that out of your brain. If you didn't know that we're talking about genetic drift, I would think to myself, this is the observation. My question would be, we started here and ended there. Is there an advantage to being red, right? My first thought would go to natural selection. And then you would run some test some hypotheses and some predictions to see if that were the case. And then once you eliminated that and you said, okay, it's definitely not natural selection, then you would know it must be by chance, which is genetic drift. Any questions about this slide? Obviously, this is just an example. I'm not going to ask you any questions about this population. And even if I were to give you this slide on the exam. There's not really any questions I could ask you. That would be too easy to ask you. Like, oh, the generation one started at 70% dominant. How did it end up? Right? That's like middle school questions. So don't expect any questions like that on the exam. Okay, here's where it gets a little bit more complicated. And you will get questions about this on the exam. You're going to need to know the difference between bottleneck effect and founder effect, both of which fall under the category of genetic drift. So I'm saying that you guys know what genetic drift is now. I'm saying now, telling you there's two different types of genetic drift. The first one we're talking about is the bottleneck effect. Bottleneck effect is the what we're going to talk about. That is not an attendance word. I know I circled it, but don't let it fool you. Anyway, again, this is another type of genetic drift. And what you need to know about it is this second bullet point right here. It results from a drastic reduction in population size. Know that for the exam. It's a nice, basic, oversimplified description that you need to know for the exam. Now, of course, when you really dig into it and start thinking scientifically, you might have to ask yourself, if a population did reduce drastically, was it by chance or was it natural selection? For example, a lot of people died when COVID-19 first hit, right? Of course, that wasn't a drastic reduction in population, but let's assume it was worse, and it was more drastic than it was. That would not be bottleneck effect, because that would be natural selection, right? Some people probably had the genome where they were less vulnerable to the virus, and other people were more susceptible to the virus. So in that case, it wouldn't be bottleneck effect. It would be natural selection. But anyway, that's digging a little bit deeper than you guys need to think about for this 100 level course. Just wanted to let you know again, we're just oversimplifying things. And for the exam, whenever it's a drastic reduction of population, you should think of bottleneck effect. But again, in real life, you should question, well, is it bottleneck effect or is it natural selection? Anyway, passing through a bottleneck, which we would consider, again, a severe reduction of population size. Obviously, it's bad because you lost a lot of your population, but that's not it. Right? It's worse than just losing a bunch of your population. And actually, in some cases, well, more so for humans than other, but sometimes populations of organisms they get too high, so nature needs to call them back. So in some, sometimes a drastic reduction can be good, but not always. But anyway, one of the reasons a drastic reduction is bad is because it decreases the genetic variability, because some alleles are lost from the gene pool. And in this case, again, it's not because it's natural selection. If you lose alleles from the gene pool due to natural selection, that's a good thing, right? So if these things start dying, whatever, I don't know, if these zebras in sub-Saharan Africa start dying, in the savannas of Africa, they start dying because they're susceptible to a certain disease, it's bad for that individual zebra, but it's good for the population of zebras because they're susceptible to the disease because of their alleles. Those alleles are now leaving the gene pool. But we're not talking about that. We're talking about genetic drift where it's completely random, right? So this results in the loss of individual variation, hence adaptability. So basically what I'm saying is it's good to have variation in the gene pool so that nature has things to choose from, right, as the population evolves. And I'll give you an example on the next slide if there's no questions about this slide. Okay. Bottleneck effect, again, they call that for a reason. It's a great visualization from the old textbook. So if you're studying it and you like the way this looks better than your textbook, download the PowerPoint. Anyway, here's the original population. It looks like a pretty simple mix, a pretty even mix, excuse me, of purple, green, and I don't know, copper, gold, I don't know what you want to call that. But then there's a drastic reduction in the population. And by the way, these are all equal other than the colors, right? So it's not like some are heavier than others, or smoother than others, or larger than others, right? They're all identical. So they all have the equal amount of chance of coming out of that bottle and into the cup. And then that's what you're left with, right? So now not only is your population much smaller, but you've completely eliminated the gold allele, and you've almost completely eliminated the orange, the green one. Not because those are bad alleles and they needed to be weeded out via natural selection, just by chance. So now look, we have less variability. We need variability for evolution, because as environments change, different members of the population will be adapted to it. Some will be more adapted, some will be less. this is how populations can survive through changes in the environment. So any questions about that side? All right, there's a real life example. I'm probably not going to ask you any questions about this, but this is not all hypothetical. This does actually happen, and it happens twice for cheetahs. The first time was during the last ice age, about 10,000 years ago. And again, I don't know. My first thought would be to think natural selection, and I would dig deeper into that because they didn't all go extinct during the last ice age, but some of them survived. But was that because they were smart enough to move to warmer spots? Or was it because there was something in their genome that allowed them to survive that? Because if so, we're talking about natural selection. But anyway, that's deeper than we need to concern ourselves with. That was the first time, and the second time was in the 1800s where farmers hunted them to near extinction, because the cheetahs are eating their livestock. But again, you might wonder, okay, well, were the hunters only able to kill the dumb ones, right? The ones that were dumb enough to be around when hunters were around, or the ones that didn't have great hearing, so they didn't hear the hunters coming, right? All those things, that would make it natural selection and not genetic drift, but we're not worried about that in this class. So again, with so little variability, they've reduced the capacity to adapt to the environmental challenges. Of course, we're not going to talk about it in this class, but cheetahs are also used as a discussion for evolution because they're so fast. And there's a concept where we talk about how like a prey will evolve. Actually, we will talk about this later. But so will the predator, right? So only the fastest of the prey are the ones who survive and reproduce. So generation after generation, they're getting faster. But then again, the predators, it's only the fastest ones that get to kill and eat. Therefore, they're the only ones who get to reproduce. Therefore, they also get quicker generation after generation. All right, so any questions about that? All right, so again, I'm not going to ask you anything about the last two extinctions. But there you go. But not extinction, but near extinctions. The next word for attendance is another birthday. His name is Donald J. Cram. And again, in a separate email for extra credit, or just DJC. You can try DJC for your attendance word. But anyway, again, for extra credit, you can look that up. Why would that person be relevant to this course? And send it in a separate email. Don't forget a link to your source. Okay, moving forward. So again, remember, we're still talking about genetic drift. Bottleneck effect is a very specific type of genetic drift. And here's another very specific type of genetic drift, and it is called the founder effect. This one, to me, is a lot more straightforward as far as what I was talking about earlier, like questioning. It's like, is it really genetic drift, or was there some natural selection in action there? This one's a little bit more like, okay, this is clearly more by chance and less natural selection. But the founder effect is exactly what it sounds like. It's when a few individuals from a larger population colonize an isolated habitat. So you have this huge population, and it's been reduced to a small population. So in a sense, it's a lot like bottleneck effect, only in that we went from a large population to a very small population. It's just in this case, it isn't because the original population died off. It's just that a small chunk of the original population left and went somewhere else. But just like bottleneck, it's just by chance, right? There was no reason that these select few started this new population. But that's how it works. And just like in bottleneck effect, because you possibly are losing some alleles just by chance, right? But it does explain some relatively high frequencies of inherited disorders that I'm not going to talk to you about, but you can look into them if you want. Look up to see if there's any more. I find these really interesting. There was I don't even remember what it was. Did I write it down? Yeah. So again, I'm not going to ask you any of these questions on the exam, but there's a African population of Dutch settlers in South Africa. They have a higher percentage of population that has Huntington's disease. That you need to know that. That's not in your textbook. That's just a good example, right? So because these are Dutch people who settled South Africa, right? It's really far away. And it's just the few Dutch people that went there. They just happen to have a higher percentage of people with Huntington's disease than the rest of the Dutch population. So now, to this day, they still have a high percentage. Pingelap people. It's a place in the South Pacific. The people who happen to colonize that tiny little island happen to have a slightly higher percentage of color blindness. So to this day, still, there's a higher population of color blindness on that island than anywhere else on Earth. Amish are a great example of that. I don't remember exactly what their ailments are. Polydactyly is one of them, where they have more than 10 digits. Because again, the people who happen to start that, right, those colonizers, they happen to have a higher percentage, and therefore to this day they still have a higher percentage. What I love about this example before I move forward is here we're talking about, you know, those Dutch in South Africa, they're really geographically isolated from where they came from, right? The Netherlands are really far away from South Africa. Pingelap, again, they're definitely isolated because they're on an island. Martha's Vineyard, I'll talk about that in a second. They're on an island. So I like that they, you know, that's easy to visualize because they're on an island. Well, these two. The Amish, I like that example because they're not on an island. You can go to Ohio and see Amish people, right? There's Ohio, there's Amish, I mean, some of them around here. There's Amish people among us. But what I love about this example is they are still isolated. They're not geographically isolated, they're just culturally isolated, right? There's not a lot of people going in or out of that community, right? So because of that, as far as genetics are concerned, they are isolated. Not geographically, but culturally isolated. Anyway, the last example that I'll give you before moving forward is Martha's Vineyard, you know, off the coast of Massachusetts. The people who founded that place happen to have a higher percentage of deaf people. Therefore, they have a higher percentage of deaf people to this day. And as a matter of fact, that's where American Sign Language was started. Anyway, if any of that interests you, if you can download the PowerPoint and click any of those links, and it'll give you more information. Any questions about that? Making good time. Gene flow. All right. This one is also in the name. Hopefully you won't have any problems understanding what gene flow is for the remembering what gene flow is for the exam. It's in the name, right? Genes are flowing. Like how else are genes going to flow other than migration, right? Immigration with an I, people are coming in, or organisms, or immigration with an E, or individuals are leaving the population. Either way, it's migration, right? Coming in, coming out, whatever. As a matter of fact, actually, I'm so glad I read this. Most textbooks do call it gene flow, but for some reason, your textbook oversimplifies it. I know Keith been saying that a lot anyway. It's a 100 level course. We're oversimplifying things. In my opinion, that's a little bit too much oversimplification. Yes, it is migration, but almost every textbook calls it gene flow. So I want you to know what gene flow is. And that's what you'll be tested on. You're going to call it gene flow on the exam. We're not going to call it migration. Anyway, again, this is another source of evolutionary change. And it's just the genetic exchange with another population. That's all it is. Sometimes you might get some new alleles, right? If you've got some individuals immigrating in, you know, immigrate with an I. Then again, you might lose some alleles if they immigrate out with an E. But generally what happens is it's going to reduce genetic differences between populations. And I'm making a completely hypothetical situation only because we talk about birds and the Galapagos Islands a lot. This is not how this worked. It's not how it happened. But imagine there was one Galapagos Island that had a bird population of finches with long beaks, really long beaks. And you had another island that had a population with really short beaks. And the islands are far enough to where they're not constantly going back and forth, but they're close enough to where they occasionally do. So when they occasionally do, that would be gene flow. And if they're going back and forth, as opposed to all one way or all the other way, that would reduce genetic differences, right? So eventually, those longer beak populations might start becoming a little shorter. And those shorter beak populations might become a little bit longer, right? Because you're reducing the differences. Any questions about that? All right, download the PowerPoint and click this link. It's another short video on evolution, including gene flow. So it's not 100% about gene flow, but it is 100% about evolution, and it also does talk about gene flow. All right, so those are the big ones. Now we'll go back and talk about the ones the biggest one, and the one we've already talked about before, but obviously it's pretty important. We're going to talk about it again. Natural selection. We're going to look a little bit closer at it. We're not done talking about it. There's still more things you need to know for the exam. Here we go. So again, when we talk about gene flow or genetic drift, right, that does not lead to adaptation, right? What leads to adaptation, the only thing that leads to natural adaptation is natural selection. Because again, it's nature selecting which individuals with all those traits that they have that help them survive, which are going to survive, right? So that leads to adaptation, because the best fit, the best adapted individuals in a population are going to be more likely to survive. Therefore, they're going to be more likely to reproduce. Therefore, that next population is going to be even better fit for that environment, right? Because the less fit, they didn't reproduce as much. So every generation will become more and more fit as more and more of those fit individuals reproduce. An example of that is the blue-footed booby. I can ask you any questions about the blue-footed booby, but we'll talk about it. It has a body and bill that's streamlined like a torpedo, which minimizes friction as it dives, because basically how it hunts, how it eats, how it goes fishing is it flies above the water, it's looking for fish. Once it sees the fish and it's close enough to the surface that it thinks it can get it, it just dives in and gets it, right? So obviously being very quick is important. So when you look at a population of blue-footed boobies, the ones that have the most streamlined, probably the ones that are going to eat the most. Therefore, they're going to be the ones that are more likely to survive. Therefore, they're more likely to reproduce. Therefore, generation by generation, they're going to get more streamlined, right? Because that's a feature that helps them survive. Another thing they have is a large tail brake, which functions as a brake. That's important, right? If you're diving in the water and you're a bird, birds can't breathe underwater, so going too deep would not be a good thing. And, right, you're streamlined. So again, the better streamlined you are, the deeper you would go, that wouldn't be good. So yeah, the large tail feather functions as a brake. And again, those birds that have that large tail feather, or larger ones, are more likely not to drown, more likely to survive, more likely to reproduce, blah, blah, blah. On a completely different note, we're not talking about natural selection, we're jumping ahead to sexual selection, but just so you know, the females prefer males that have bright blue feet. That is sexual selection, not natural selection. But in my opinion, those things overlap a lot. Because even though the females aren't thinking about it, they're attracted to these bright blue feet. But what's happening on the natural selection side of that is it's the healthiest birds, the healthiest males that have those bright blue feet, right? Because they're not sick and they're well fed. So they're good breeding stock. So even though, again, it's not a conscious decision where the female's like, no, that's going to be good for our gene pool. But they don't think that. They're just attracted to bright blue feet. That's sexual selection. But I'm just saying, even though you don't need to know it for the exam, just so you know, there's a very close relationship between sexual selection and natural selection. Anyway, if you download the PowerPoint, you can watch this video. There's another one about natural selection. Are there any questions about this slide before I show you a picture of a blue-footed booby? There it is. Streamlined body, streamlined bill, big old tail feather. There's a brake, bright blue feet to get the ladies, so to speak. Right? It's a male blue-footed booby. All right. So I've already used this word a lot, but now let me officially explain what it means. The word is fitness, specifically relative fitness. Relative fitness is the contribution an individual makes to a gene pool relative to the contributions of other individuals. And again, like some people say, and I don't really like the term, but survival of the fittest. And again, the reason I don't like that term, survival of the fittest, is because survival is only half the story. But yeah, if you survive, then you're obviously more likely to contribute more to the gene pool because you're alive and you reproduce. So that is relative fitness. So when I say something is fit, I'm saying there's something about it that's going to help it survive and help it reproduce. Some things. So there's got to be a whole mixture of things, a whole slew of phenotypes that make it fit for its environment. A good fit for its environment. It's adapted to its environment. Like if you put a blue-footed booby in a forest, it's probably not going to do very well, right? It doesn't know how to catch food to eat there. It's not adapted to a forest. It's adapted to flying over oceans and diving in and getting its food that way. So any questions about this slide? Okay. Next word for attendance is a date. Not a word at all. 1970. Just write down 1970 or a year. But again, for extra credit, you want to send me a separate email. Tell me what happened on this day in 1970. That would be related to this course, or my courses, including environmental biology. All right, if there's no questions, we will move forward. We are making great time, guys. But again, I'll remind you, if I'm going too fast, slow me down. But also, if I'm going too fast, remember they are being recorded, so you can always go back and rewatch the lecture. All right, let's talk about the 3 general outcomes of natural selection. I always thought this was a simple topic, but sometimes people have a hard time with it. Everybody thinks differently. We're going to use an example of mice. I'm not going to ask you any questions about mice, but we're going to use them in this example to explain the 3 different outcomes. If we were to graph the coat color of a population of mice, we would get a bell-shaped curve. I'm going to explain it when I show you on the next slide. It'll really make more sense. And if natural selection favors certain colors over others, the population will change over generations. And then we have 3 general outcomes. Hold on, really quickly. Yeah, I have my slides backwards, so here we go. This is what I'm talking about, right? This is the original population. We have a whole population of mice, some of them really dark, some of them really light, but a majority, right? Most of the population is middle, right? We have a lot of in between, a lot of that average color. Very fewer where it's slightly darker and slightly lighter, and even fewer where they're really dark or really light, right? So that's a bell curve, right? So, hey, that would be a good test question too, just to remind you, even though you've already taken the exam. The fact that it's a bell curve, that would be a good example of pleiotropy, right? Because we have a lot more polygenic inheritance, because we have a lot of genes contributing to that one trait. Hence the bell curve. Anyway, so this is our original population, right? And the 3 different outcomes, as evolution changes them, is we have directional selection, which means it's going one way or the other, right? In this particular example, it's going this way, right? So we're having more dark, right? But we still have that bell shape. It's just now we're going to have, the dark ones are going to be a little bit more common, and the light ones are going to be a little bit less common. It's shifting to the right. Of course, that's just an example. It would have been equally as valid if it shifted to the left. The point is, it shifts in one or the other direction. Does that make sense? That is called directional selection. It's in the name. I hope it makes sense. So let's assume these mice live on a volcanic island. I don't know, maybe in Iceland, right? It's volcanic, but it's still cold. Now let's say things started to warm up. So we had less ice and less snow. Because of that, these old dark mice could hide amongst that dark volcanic rock easier and hide from predators. Therefore, the population would shift that way. Or let's say the opposite. Again, directional selection. Let's say we had a mini ice age. And now all that dark volcanic rock is starting to get covered more with snow and ice. So now those black ones, those dark ones are starting to stick out more. So in that case, again, we would have directional selection. We'll just go the other direction. All right. That one's pretty simple. I'm going to skip ahead to C before we talk about B. The other one is called stabilizing selection. Stabilizing is when you have fewer and fewer of the extremes, but more of the average. Okay, so again, the darker ones in that situation, they're dying off. The lighter ones are dying off, but for some reason, the gray ones are at an advantage. That's pretty simple too, right? Stabilizing. The opposite of stabilizing is disruptive selection. This is when you get fewer and fewer of the average, but you get more of the extremes. More dark, more light, fewer gray. There's a real life example of this that I like to talk about. The unofficial name, they call it the sneaky bastard. I think it's called the sneaky bastard. Anyway, there's a population, a species of beetles, and they fight, right, to mate. They fight each other, and the winner gets to mate with the female. So what happens is, obviously, the bigger, stronger males, those are the ones who win, and they get to reproduce. So you're thinking, hopefully, you're thinking to yourself, yeah, dude, that sounds a lot like directional selection, right? Because the bigger ones are going to be more common, and the smaller ones are not. But here's where the sneaky bastard comes in. What happens is, while the big males are fighting, the really small males that look like females just walk right past them, and the fighting males don't even notice it's a male. So that small male goes in there and mates with the female while the other two are fighting, and then he wins the day. So because of that, you're getting a lot of the small beetles because they're sneaky. You're getting a lot of the big beetles because they're winning. But those in between beetles, not only can they not win a fight, but they can't sneak past and fight in big fighters. So anyway, any questions about that? Ooh, Great time. We're making great time. Next thing we'll talk about is sexual selection. Sexual selection is when individuals that have certain traits are more likely than others to obtain mates. We also call this intra-sexual competition, meaning members of the same sex, usually males, are competing against each other to choose a mate. Intersexual selection, interselection of mates. And this leads to sexual dimorphism. You know what that is for the exam. There might be one question where I ask you what that is. That is the distinction in appearance between males and females. And it's not necessarily directly associated with reproduction or survival. Directly. No, remember, I was just talking about that blue-footed booby. So not directly associated with the survival, but again, it's the healthier males that have those brighter blue feet. So in a sense, they are related. We have less than a minute. So when we get back, we'll finish this discussion about sexual selection. But I'll remind you, your first attendance word was to tell me your thoughts on meeting somewhere else for a lecture. Yeah, I'll probably ask again on Friday too, but I'll pose it in a different way. You'll see what I mean when we get there. So don't forget to ask that question for your first attendance word. And don't forget to ask me one question about the material covered for your last attendance word. And that's it. See you either on Friday, or if you want to come to my office or meet me along your line, I will see you for labs day if you want. Again, you don't have to. I'm trying to make things a little bit easier here towards the end. For those of you who are online, don't forget to send those emails, please.